Hey, my name is Madeline Sesser, and I'm going to talk a little bit about my lab final for microbiology. Due to COVID, we did a online virtual lab on ViewMe, and for our final, we did a Save the Patient simulation, which is where we go and we do what everyday medical professionals do. Someone is sick, you need to figure out what is making them sick, what bacteria they have. So you run some tests so that way you can find out which antibiotic to use so that they can get better. So we started out with the option of 12 microbes, and then we had to narrow it down to 1. And the first thing that we had to narrow them down was called a gram stain. It is where you can stain bacteria, and it helps differentiate between whether it is gram negative or gram positive. So this is done in 4 steps, and you have a crystal violet dye that goes on, and it stains all the bacteria purple. And then you have an iodine that goes, well, then you rinse with water, and then you have an iodine that goes on the slide. And what it does kind of is break down the cell a little bit, like the cell wall of positives, of gram positives, and this allows the crystal violet to attach really well, and so they turn purple. But it does not break down the cell wall of the gram negatives. And so then you put some type of decolorization on it, like alcohol, and it strips all the negatives of the purple stain. And then you add saffron, which gives a pink stain to the gram negatives. So you end up with the possibility of pink and purple, purple being gram positive and pink being gram negative. On my slide, I had purple, so I was gram positive. So that broke it down, and then it left me with 6 options of bacteria that I could possibly be. And this helped to determine which test to do next, because you need to do certain tests for gram negatives and certain for gram positives. So the next test I decided to run between the 6 was a catalyst test. I had done a little research on my bacteria, and I knew 3 would be positive for the test and 3 would be negative, which was a great way to break down more and give me a smaller option. So for this test, you have a streak of your culture on a plate, and you drop hydrogen peroxide onto the culture. And what it's doing is it's looking to see if the bacteria has the catalyst enzyme that destroys hydrogen peroxide. If it bubbles, it has the enzyme. If it does not bubble, then it does not have the enzyme. I actually had one that was negative for the catalyst test. There was no bubbles. It did not destroy the hydrogen peroxide. So that left me, that broke it down, and it gave me 3 options instead of 6, because 3 were positive and 3 were negative. So now I had 3 bacteria that I could choose from. And through a little bit more research, I was kind of nervous on which test to run. I wasn't really sure, but I went ahead and I chose the blood hemolysis, excuse me. And this sees if the microbe has a hemolytic enzyme that basically destroys, it can just destroy, or it can destroy and digest red blood cells, more or less. And what you do is you have a agar, blood agar plate, which I had a 5% sheep blood plate, and you just streak your culture onto your plate, and then you incubate it overnight. And when you take it out, then you can see the results. If there is nothing changed, then that would be gamma, and that means that it has no enzymes at all that do anything to blood cells. Like, they just don't worry about them. And then if it is alpha, it is slightly discolored. It means it had a little effect. It doesn't really digest them or anything. It may destroy them a little bit, but it's just a green like schmear, kind of like it's just a little discolored. And then beta, it actually has halos, or it is completely cleared and you can see the bottom of the plate. And my enzyme my microbe I had halos and I could see the bottom of the plate. And actually at this point in the View Me simulation, I managed to somehow forget to recap my plate before I put it in the incubator, and I'm not sure if it just caused problems with the outcome or if it caused problems with the next test. But after the blood chemolysis, I decided to do a mannitol fermentation test, mannitol salt agar. And this determines if the microbe can use mannitol as its main source of carbon. And then it also tells if it's salt sensitive or not, because you are using a mannitol salt agar. So what you do is you inoculate a tube of the mannitol salt agar with your culture, and you incubate it overnight, and then when you pull it out, it will either have a color change or no color change. If it has a color change, that means that the pH has changed from the microbe using the mannitol as a carbon source and fermentating. If there is no color change, then the microbe did not use the mannitol and it is negative. My test ended up being no color change, red. Because of this, I chose E. avium as my bacteria. Now, whether or not this is accurate, I'm not sure, because I did have a fault in my exam and I forgot to recap my items, my plate, before I inoculated it. And that's one of the things that you have to do on every test. You should be flaming your tubes, you should be flaming your loop or your wire. You should be flaming tubes before and after inoculation. You need to recap immediately or it can cause contamination. Flaming your loop and wire before and after. None of my tests had reagents. Well, I guess the catalyst is considered a reagent because you use hydrogen peroxide to drop it. And hopefully I got it right. I did not save my patient, unfortunately, because I left the cap off. I was not able to choose the correct bacteria. But other than that, I think everything went fine, and had I put that cap on, then I would have been able to have the correct bacteria. But this was definitely a good lesson, learning. I do wish that we could have done this on in class, hands-on. This was a learning curve for sure to figure out a virtual lab. It was fun, it was interesting, and definitely learned to make sure I cover the basics like flaming and putting caps on. So, thank you so much.